this video we're going to look at the Nobel Prize in Physiology or Medicine from the year 1908, awarded to Ilya Mechnikov and Paul Ehrlich in recognition of their work for immunity. More about that later, of course. First on Ilya Mechnikov, of what he must work. There's a little bit more about why he got the prize. During the second half of the 19th century, it was clear that many diseases are caused by attacks of microorganisms. It was also determined that our immune system protects us against these attacks. Ilya Mechnikov contributed in several ways to our understanding of how this happens. After studies of starfish larvae in 1882, he pointed to phagocytosis as one of the immune system's ways of operating. By this he meant that certain cells in the blood, white blood cells, work by encapsulating and destroying harmful bacteria and other microorganisms. Here you can see that this is coming from the official Nobel Prize site. Here you see the man at work in his laboratory. Then some biographical information, also coming from the official site from the Nobel Prize, summarized by me. He went to Messina to work on comparative embryology. He discovered the phenomenon of phagocytosis. The discovery was made when Mechnikov observed in the larvae of starfishes mobile cells, which might, he thought, serve as a part of the defenses of these organisms, and to test his idea, he introduced into them small thorns from a tangerine tree, which had been prepared as a Christmas tree for his children. Next morning he found the thorns surrounded by mobile cells, and knowing that when inflammation occurs in animals which have a blood vascular system, leukocytes, white blood cells, escape from their blood vessels, it occurred to him that these leukocytes might take up and digest bacteria that get into the body, the same as they digest the thorns. Ultimately, in 1883, Mechnikov gave at Odessa his first paper on phagocytosis. Apart from its fundamental importance in immunology, the discovery has a marked influence on Mechnikov himself. Some proof of it he found in the small freshwater crustacean diphtheria, in which he found that several fungal spores which attacked it were themselves attacked by the phagocytes of the crustacean. He then studied anthrax bacilla and found that the more virulent strains of these were not attacked by the phagocytes, while the less virulent strains were. At the Pasteur Institute in Paris, Mechnikov was engaged in work associated with the establishment of this theory of cellular immunity, which, like many great advances in science, encountered considerable hostility. Well, this is some biographical information summarized by me. If you want the complete biographical information from the official site, then you have to follow this link, type it over or something. Here again you see the man at work in his laboratory, dans sa laboratoire. That's French. Now we go to the second laureate, Paul Ehrlich. You see him at work in his office in Frankfurt. First, from the official side, some of his work. Our immune system protects us from attacks by microorganisms. As part of its defenses, the immune system forms antibodies in the blood that neutralize poisons or toxins that are formed by bacteria. One of Paul Ehrlich's contributions to immunology was the transfer of blood serum with antibodies to treat and counteract diphtheria, which he carried out with Emil von Behring, also a Nobel Prize winner. Ehrlich speculated that cells have a kind of receptor that binds to the harmful substances. The receiving elements are knocked off and the cell become antibodies. This is again coming from the official Nobel Prize site. You see the link here. There's some biographical information. He did further, that's Ehrlich of course, important work on immunology, especially on hemolysins. He also showed that the toxin-antitoxin reaction is, as chemical reactions are, accelerated by heat and retarded by cold, and that the content of antitoxin is in antitoxic sera varied so much for a various reason that it was necessary to establish a standard by which antitoxin content could be exactly measured. This he accomplished with von Behring's anti-diphtheric serum and thus made it possible to standardize this serum in units related to a fixed and invariable standard. The methods of doing that Ehrlich then established formed the basis of all future standardization of sera. This work and his other immunological studies led Ehrlich to formulate his famous side chain theory of immunity. He now devoted himself to chemotherapy. His aim was, as he put it, to find chemical substances which have special affinities for pathogenic organisms, to which they would go as toxins go to toxins, to which they are especially related, and would be, as Ehrlich expressed it, magic bullets which would go straight to the organisms at which they are aimed and destroy them, of course. To achieve this, Ehrlich tested hundreds of chemical substances selected. He studied, among other subjects, the treatment of trypanosomiasis and other protozoal diseases and produced trypan-red, which, as his assistant Shiga showed, effective against trypanosomes. He also established with A. Bedheim the correct structural formula of atoxyl, the efficiency of which against certain experimental trypanosomiasis was known. This work opened the way of obtaining numerous new organic compounds with trivalent arsenic, which Ehrlich tested. At this time, the spirogate that caused syphilis was discovered by Schaudin and Hoffman in Berlin, and Ehrlich decided to seek a drug that would be effective especially against this spirogate. Among the arsenical drugs was one, the 606 of the series tested, what a work, which had been set aside in 1907 as being ineffective. But when Ehrlich's former colleague Kita Sato sent a pupil of him named Hatta to work at Ehrlich's institute, Ehrlich learned that Hatta had succeeded in affecting rabbits with syphilis and asked him to test this discarded drug on the rabbits. Hatta did so and found that it was effective, very effective. When hundreds of experiments had repeatedly proved its efficacy against syphilis, Ehrlich announced it under the name salvasan. Eventually, it turned out that the 913th arsenical substance to which the name neosalvasan was given was, although its curative effect was less, more easily manufactured and being more soluble, became more easily administered. Ehrlich had, like so many other discoveries before him, to battle with much opposition before salvasan or neosalvasan were accepted for the treatment of human syphilis. But ultimately, the practical experience prevailed and Ehrlich became famous as one of the main founders of chemotherapy. Again, this information is from official Nobel Prize summarized by me. If you want to read the complete information, then you have to follow this link. Here again, you see the man at work in his laboratory. And we go to the award ceremony speech by Professor Count Murner. Some time ago in this place, a short description was given of the development of medicine. In it was emphasized that medical science today has set itself the task of attempting to prevent disease. In order to achieve this aim, one must attempt, on the other hand, to find the disease germ and destroy it, and on the other hand, to give the body the strength to resist attack. As regards the latter, the possibility of obtaining protection against diseases has now been known, for the observation has been made that in many cases organisms which have gone through an infectious disease will acquire protection against being attacked by the same disease again. We then say that organisms have acquired immunity against the disease in question. In scientific development, however, there is a very big step between this observation and the knowledge of the changes which have taken place in the organism through immunization, and it's also a very big step from the same observation to the ability, conscious and without the danger attendant upon the course of the illness, to give the organization such power of resistance. It was therefore rightly considered to be an epoch-making and blessed moment in the history of medicine when Edward Jenner introduced, more than 100 years ago, now 200, protective vaccination with cowpox substance, which can give immunity against a disease, namely smallpox, the ravages of which the present generation can hardly imagine. Great though the practical importance of Jenner's discovery was, it did not advance the development of the study of immunity in respect of other diseases or permit any deep penetration into the problem of immunity generally. The prerequisites for such a successful scientific elaboration of the study of immunity were still missing. The first and most important condition for making the problem of immunity the subject of real scientific research was namely to establish the cause of the disease. It was the revolutionary work of Pasteur and Koch, which was done approximately 3/4 of a century after Jenner's discovery, which laid the necessary foundation for the present important development of the study of immunity. Ilya Mechnikov was the first to take up consciously and purposefully, by means of experiments, the study of the question so fundamental to the question of immunity. By what means does the organism vanquish the disease-bearing microbes attacking the organism in which they have succeeded in establishing themselves and developing? At first, his experiments were restricted to the lower animals. This was the case in his important work concerning a kind of infection in certain microscopic aquatic animals, the so-called water fleas. If the guiding principles behind these investigations were not known, they could appear to be remote from any medical interest. They were, however, the first links in the chain of investigations leading to the phenomena of immunity, also in mammals and in humans. These investigations opened the way for Mechnikov's theory of phagocytosis. According to this theory, the microorganisms are destroyed by the activity of cells in the organism. Certain kinds of cells in the body of humans and animals, namely the supposed to have, in addition to other function, the task of catching and destroying disease-producing microbes which have succeeded in penetrating the organism, and also of rendering certain bacterial poisons harmless. I cannot here give a report of the comprehensive work and valuable observations which go to make up this theory of phagocytosis, but an important aspect of this research is that it makes a special study of certain types of cells, and that first of all the importance of the cells for the phenomenon of immunity is emphasized. One can safely predict that even other features are of more immediate importance in this doctrine. Nevertheless, the abundant actual observations which have been made with regard to the importance of the cells to the problem of immunity will always remain of great and permanent value. In the doctrine of immunity, as in other provinces of biology, the activity of cells, which are considered as being the focus of organic life, remains a factor of the highest importance. The research of recent years into the question of immunity has thrown the importance of Mechnikov's work into strong relief. As recognition of Mechnikov's accomplishments in initiating modern research into the question of immunity, the direction and development of which, particularly in his early stages, he profoundly influenced, the Caroline Institute wished to honor him with this year's Nobel Prize. Like other biological processes, the phenomena of immunity are of a very complicated nature and provide a field of research of almost unlimited extent. It's obvious, therefore, that this field can be approached from various directions. Recently, other comprehensive and equally successful research into immunity has been conducted, only certain parts of which touch upon the theory of phagocytosis. I will attempt to explain these in a few words. It has been shown that protection against disease can be of two kinds. It can consist in the ability to destroy microbes or to inhibit their further development. This is a bacteria-destroying immunity. But there's also a protection of another kind, one which acts against the bacteria products. The damage which is the disease-producing microorganism cause, namely, is conducted by the poisons which these organisms produce and which are distributed into the blood fluids, by the blood fluids. A certain kind of immunity occurs against this danger as well, the so-called poison immunity. The The best known example of this is the use of anti-diphtheria serum, when through a serum injection substances are introduced into the organism which act as antitoxins against diphtheria poison. It has been discovered that poisons which are produced by bacteria have the property, as have many other substances also, of causing the production of elements in the organism, which has an antagonistic effect especially and exclusively directed against the subject which causes the production of the elements. This we call the formation of antibodies. After immunity has been achieved, such antibodies are found in the humor of the organisms. Furthermore, it has been possible to show that these antibodies are of great importance not only as regards protection against disease-producing microorganisms themselves, but also, above all, as regards protection against the toxic products of these organisms. An endless series of questions now arises. Why are antibodies only built up against some substances and not against all substances which are foreign to organisms? Where are antibodies formed? By what process are they formed? What is the nature and constitution of these antibodies? How do they react on the microorganisms and the poisons? And various other questions which are important as regards the development and practical utilization of the theory of immunity. It's also a matter of great interest that connecting links have been found between the theory of immunity and the normal physiological processes. A great deal of intensive and very fruitful work has been devoted to this question in the last 1.5 decades. A large number of research scientists have served the cause of science well by their discoveries and its achievement. It's not possible here to report on the extent to which the questions have been answered, neither is it possible to describe the separate accomplishments of scientists in this field. Well, this is a summary of the ceremony speech. If you want a complete summary speech, then you have to follow this link, type it over or something. And there's even more information in the speech read section of the 1908 Nobel Prize for Medicine or Physiology, which you can access via this link. And last, the Nobel Prize diploma awarded to Paul Ehrlich. Mechnikov must have received a similar but not identical Nobel Prize diploma. That's usually how it works. And they're always beautiful. Next week, we will go into this Nobel Prize once more with the Nobel Prize lectures given by Mechnikov and by Ehrlich. For now, thank you for watching and maybe till another time.